Good day, beautiful people. I hope you're good. I'm doing great. I am Hanifate Abelia, one of the member of Group 5, and we're going to discuss the Lesson 5, the aspects of gene therapy. So the other members are Agot Sandria Jean, Hirda Kiana, Bronyola Nico, Sayed Ryan J, Guillermo Debril, Balbuena John Marl, and Kachela Robinson. So let's start. The lesson objectives is at the end of this lesson, the student should be able to describe gene therapy and its various forms and assess the issues, potential benefits, and detriments to a global health. So first, let us know what is the meaning of gene therapy. Gene therapy involves altering the genes inside your body cells in an effort to treat or stop a disease. Genes contain your DNA or deoxyribonucleic acid. Then the introduction. Medical science has detected many human diseases related to defective genes. This type of diseases are not curable by traditional methods like taking readily available medicines. Gene therapy is a potential method to either treat or cure a genetic-related human illnesses. So as you can see here, this is the human diseases, and so there are a lot of human diseases, and this type of diseases are not curable by traditional methods like taking readily available medicines. The traditional medicines refers to health practices, approaches, knowledge and beliefs incorporating plant, animal, and mineral-based medicines, spiritual therapies, manual techniques, and exercises applied singularly or the combination to treat, diagnose, and prevent illnesses or maintain well-being. A team of researchers at the Harvard Medical School in 2015 and the Boston Children's Hospital. So this is the Harvard Medical School and the Boston Children's Hospital. Stated that they were able to restore basic hearing in genetically deaf mice using gene therapy. The research team also reported that they have restored a higher level of hearing down to 25 decibels, which is actually equivalent to a whisper. The musculette's eye and ear was identified as ANC80, which enable the transfer of genes in accessible otor hair cells when introduced into the cochlea of the year 2017. In 1971, human gene therapy was actually first realized when the first recombinant DNA experiments were planned. It can be simply viewed as insertion foreign DNA into a patient's tissues that hope to successfully eradicate the targeted disease. It occurred over the last 20 years was actually inspired by success of recombinant DNA technology. Without a doubt, gene therapy is the most promising, yet possibly and favorable medical field being studied.
Good day everyone, my name is Robinson M. Kachela and I am going to report the basic process in Lesson 5, the aspects of gene therapy. The basic process, replacement of mutate gene that causes disease with a healthy copy of the gene. Inactivation of a mutate gene that is functioning improperly. Introducing a new gene into the body to help fight a disease. In general, a gene cannot be directly inserted into a human gene or cell. A gene is inserted into another gene using a carrier or vector. At present, the most common type of vectors are viruses that have been genetically changed to carry normal human DNA. Viruses have evolved a way of encapsulating and transporting the genes to human cells in pathogenic manner.
Good morning, everyone. My name is Kiana Dihuda, and today I am going to discuss my topic, which is all about gene therapy. So, what is gene therapy? So, gene therapy is based on correcting a disease at its root. So, it is a way in fixing the abnormal genes that appear to certain diseases. So, from the word itself, gene, meaning it is hereditary. And from the word itself, therapy, meaning it is something that is used to cure or to treat any certain diseases. Now I will be talking about the two types of gene therapy. The first type is the somatic gene therapy. So the somatic gene therapy is the manipulation of genes in cells that will be helpful to the patient but not inherited to the next generation. So this is according to Namser-Gurn, 1988. The second one is the germline gene therapy. It involves the genetic modification of germ cells or origin cells that will pass the change on to the next generation. Origin of cells that will pass the change on to the next generation. So the difference between these two types of gene therapy is that the somatic gene therapy is the manipulation of genes in cells which is not inherited to the next generation, while the germline gene therapy is the modification of germ cells and it will pass the change on to the next generation. So somatic, it is the manipulation and it will not pass into the next generation, while the germline is the modification and it will pass into the next generation. So to add more about these two types of gene therapy, the germline gene therapy is when DNA is transferred into the cells that produce reproductive cells. Eggs, may it be eggs or a sperm cell. So eggs are for girls and sperm are for boys. So while the somatic gene therapy is when DNA is transferred into body tissues. So what are these body tissues? For example, the muscle tissues, our nervous tissues, and more. So that's it for my discussion, and now I'm going to hand you over to the next reporter. Thank you.
My name is Janmar Balbuena, reporting stem cells therapy. Stem cells therapy are mother cells that have a potential to become any type of cells in the body. One of the main characteristics of the stem cells is their ability to self-renew or multiply while maintaining the potential to develop into other types of cells. Stem cells can become cells of the blood, heart, bones, skin, muscles, brain, among others. There are different sources of stem cells that all types stem cells have the same capacity to develop into multiple types of cells. Stem cells are derived from different sources, two of which are embryonic.
Hi everyone, my name is Deborah Hermo. I continue stem cell therapy. The embryonic stem cell are derived from a 4 or 5 days old human embryo that the phase of development. The embryonic stem cell mo derived to 4 or 5 days human embryo that in parasitic phase. Bago sila ma-develop. The embryo are usually extra that have been created in IVF means in fertilization clinics where several eggs are fertilized in a test tube, then implant into woman. Kostra 2013. The somatic stem cells are cells that exist katuning mga cells na exist throughout the body after human development, after embryonic development, and are found inside of different types of tissue. Natin mafound doon na lalahig kind of tissue. This stem cell have been found in tissue such as the brain, bone marrow, blood vessels, skeletal and muscles, skin and the liver that remain in a non-dividing state for years until active by decrease or tissue injury. These stem cells can be generated, arranged. Kani ko ng stem cell kaya mo generate as a range of cell. Apart from originating organ, even the entire original organ. It is a general truth. Adult somatic stem cells are limited the ability to differentiate based on the tissue of origin. Based on mga tissue of origin daw. But it is some evidence suggests that they can differentiate become other cell types. Costa 2013.
Good day everyone, my name is Ryan J.G. Sayod, and today we're going to talk about bioethics. So, before we begin talking about bioethics, we should know what bio and ethics means. So, bio means life, our human lives, animal lives, and even environmental lives. Ethics means the moral principles that governs an individual's behavior. It decides if an action is morally right or morally wrong. Now let's proceed to bioethics. Bioethics is the study of ethical issues emerging from biological and medical advancements, questions, issues, and even controversies, like is it right if we use a living being as a subject to test if a newly developed medicine is working? From there, more questions will pop up, such as questions concerning its side effects to the subject. There are a lot of things to know about a newly developed medicine, but I think first we should know if actually using living beings to test a newly developed medicine is morally right. Bioethicists. These are the professional individuals that study and conduct research concerning unethical, social, and legal issues arising in biomedical research. Now let's talk about the relationship between bioethics and bioethicists. Bioethics establishes a mutual understanding from multiple perspectives. It uses its way to give an equal and fair consideration regarding issues that concern human, animals, and environmental rights. It helps us know that it's not just your moral question regarding another case of medical or biological advancements, and it's lets your questions with others be voiced out. Meaning, it's not just you. There are a lot of people who are question about if it is morally correct or not morally right. Now, bioethicists, these are people, the one who studies the questions and look for an answer, as they are concerned with ethical questions that appear in the relationships among sciences, biotechnology and medicine, politics, philosophy that concerns with life. Now, let's talk about the 4 principles of bioethics. First one is autonomy, beneficence, non-maleficence, and justice. Now, autonomy is the right of an individual's right to make his or her own decisions that concerns with their own health, and that right should be respected. Beneficence. It states that healthcare providers must strive to improve in order for their patients to benefit even more. Now, non-maleficence. In accordance to beneficence, healthcare providers must not harm their patients as well. And lastly, justice. As well as in accordance to beneficence again, patients must share an equal cost for the benefit they receive from the healthcare system. That is all for my report, and the rest will be for the next reporter.
Good day everyone. I am Nico Bruniola from Group 5 and I'm about to report the scope of bioethics.
and the five major issues. So what is scope of biotics? It is artificial reproductive technologies. It has stem cell research and human genome project. It has a possibility of successful reproductive cloning, and it has synthesis of new life forms and pre-implantations of genetic diagnosis. So let's go to the five major. Let's go to the five major issues. So what is five major issues? Five major issue means important or serious topic or problem for discussion. So this is the five major issues. First one is abortion. Abortion is a removal of fetus or embryo from a woman's uterus that results in a termination of pregnancy. And the second one is euthanasia. Refers to the act of purposely ending a life to eliminate untreatable suffering. The third one is eugenics, a type of DNA manipulation that creates selective breeding to improve the human race. And the fourth one is human cloning, the scientific production of an identical human cell, tissue, or an even entire body. And the last one is the stem cell research, the area of research that studies the properties of stem cells and their potential use in medicine. So that's all for my reporting. God bless all, and thank you.
So, good day everyone. I am Agot Sandrajin, and I'll discuss the summary of Lesson 5, the aspects of gene therapy. So, gene therapy is a method that may treat or cure genetic related human illnesses. There are two forms of gene therapy. One is a somatic gene therapy, which involves the manipulation of genes in cells that will be helpful to the patient but not inherited to the next generation. So, again, only the patient has the advantage of this therapy. And the other therapy is the germline gene therapy, which involves the genetic modification of germ cells or the origin cells that will pass the change to the next generation. So germline therapy is when DNA is transferred into cells that produce reproductive cells, eggs or sperm cells in the body. This type of therapy allows for the correction of disease-causing gene variants that are certain to be passed down from generation to generation. So both the patient and the next generation has the advantage of it. So there are many ethical issues on the gene therapy. Some of these issues are about the questions on whose authority or power to decide which common traits should be altered. Other concerns are on the discriminatory effects of those who may not or cannot avail gene therapy. For example, the somatic mutations. It occurs in non-germline tissues and it cannot be inherited. So as I have said, only the patient has the advantage of this therapy. While the germline mutation is present in egg or sperm cells and it can be inherited. For example, class cancer family syndrome. So since the parent had already done therapy of this, so since it is present in egg or sperm cell, it is heritable by the children or the child. So all cells affected in upstream of the child. So again, both parent and the next generation or the children or the child, it has the advantage of it. So that's all, And thank you for listening. Have a good day.